Hello and welcome. We're going to talk in this section about genome informatics. We're going to first here in this video introduce genome informatics. What is a genome? How do we sequence it with so-called first, second and third generation, if you will, sequencing? And then what can we do with a genome sequence once we have it? And then in our next video in this section, we're going to actually talk about workflows for next generation sequence analysis. We'll begin with RNA-seq analysis, or the analysis of expression data via this direct sequencing kind of approaches. So a genome, of course, is the total genetic material of an organism by which individual traits are encoded and controlled and ultimately passed on to future generations. Recall, of course, that we're a mosaic of the genetic material from our ancestors, half from mum and dad, right the way back through the generations. And this, of course, motivates genetics and the study of traits and variations in genes. And genomics is indeed intimately related and, in fact, underlined by genetics, but it is distinct. Genetics I think of as primarily the study of individual genes or families of genes, mutations within those genes, and their inheritance patterns in order to understand specific traits, for example. So genomics really expands upon this classical genetics approach and considers aspects not just of individual genes, but of entire genomes. And because of this large-scale nature, it nearly always requires computer-aided approaches. That's a big defining aspect of genomics. Genomes, of course, come in many different shapes, and as we'll see in a moment, sizes too. They're primarily DNA, but they can be RNA, in the case of viruses like the coronavirus, for example. Some genomes are circular, others are linear. They can be organized into discrete units, like chromosomes, so-called because when they were first visualized all those years ago, they absorb dye really well, right, the chromosomes. Or they can be freestanding molecules like plasmids and the like. Now, viruses have the smallest genomes and eukaryotes the largest. Humans actually are reasonably far from the top of my little log plot here, where I've plotted the number of protein coding genes on that vertical axis versus overall genome size and megabases down here in the bottom in this log scale. Humans, we have just over 20,000 protein coding genes. We have a genome size of about 3.2 billion base pairs. So it's a little bit humbling looking at these sorts of plots and where we are in them. But genome sizes, of course, for down the bottom here, we have bacteriophages and viruses that can range from a few thousand bases to several hundred kilobases. Bacterial genomes, from about half an MB to 10 MB. Then eukaryotes, right at the top here, that are pretty diverse. They range from about 10 MB in some fungi, for example, to up to 100 MB in certain flowering plants that are the species up at the top of this plot here, for example. Now, the data for that plot, I obtained it pretty quickly from the NCBI Genome Database shown here behind me, and we have, of course, used NCBI databases and tools pretty substantially by this stage of the course. We're well aware and well familiar with these resources. However, NCBI Genome Database, this one behind me here, it's not as popular in the field as some other genomics resources. Most notably, we have Ensemble. This is actually based out of the EBI, the European Bioinformatics Institute, which again, we're quite familiar with the EBI. This is Ensemble, and we'll get to use it in our hands-on session together with this one, a very popular resource, bioinformatics resource and genome browser resource at UCSC, the University of California, Santa Cruz, our sister institute just up the coast here. Now, we'll get the opportunity to use both of these, as I said, in our hands-on section this week, really delving into analyzing genomics data and workflows for NGS analysis. So we'll come back to these and talk about what makes them good compared to other databases, for example. Now, just like every talk on introductory sequencing or genomics, you must have a slide on the father of this field, Fred Sanger. He's, of course, considered the father of genomics. He was the originator of Sanger sequencing, or chain termination sequencing, back in the late 1970s, for which he was awarded the second of his 2 Nobel Prizes. The first one was for protein sequencing, of course, before this one. And I've rather than delving into the details of Sanger sequencing, which many of us will have explored in other classes in the past, I've placed a link on our class website to a very nice video by Jonathan Wiseman from UCSF that really delves into different sequencing approaches from Sanger to capillary to shotgun sequencing, so-called next generation sequencing, and indeed third generation sequencing. And I don't want to spend too much time on these methods here, but what I will mention here is that Sanger sequencing, good old Sanger sequencing, the reads were comparatively long. They were in order of 750 to 1,000 base pairs, right? So much longer than the 300 base pairs we get out of a modern Illumina sequencer, for example. And they had comparatively low error rates, right? 1 in 10,000 bases here, for example. Again, better than many other modern approaches. Now, the problem with Sanger sequencing, or indeed capillary sequencing, is scaling up, right? And the cost that's associated with that and the time it would take to do this on large genomes and many, many genomes. It's just not feasible to do that. It was back in the 70s, right? The first genomes sequenced using these approaches were pretty modest. Phi-X, for example, bacteriophage, completed back in 1977. It had 5,386 base pairs of single-stranded DNA, only 11 genes. And the second here, Haemophilus influenzae, that I'm showing, completed in 1995. It had under 2 million base pairs of double-stranded DNA this time, about 1,740 genes. Notice the date here, that's 1995 here, so keep that in mind, because before that was finished, in 1990, the Human Genome Project was initiated, a really ridiculously ambitious project at that time. There was a lot of controversy, a lot of discussion within the community about what this large-scale project would really entail and what it would take away from traditional kind of hypothesis-based biomedical research that was and still is the go-to research model for funding in the United States and elsewhere. It was originally led and pioneered by the ever controversial James Watson, of course, of Watson and Crick fame, and later of fame for other less wholesome reasons, of course. He was fired 2 years in, and subsequently, there was a year of interim leadership, and then Francis Collins was recruited from the University of Michigan, right? The cystic fibrosis gene discoverer, of course, famous scientist, really working scientist at his time, who's now head of National Institute of Health as a whole, of course, the main funder of biomedical research in the United States. But there were many other superstar geneticists, leading lights in this project, including like Eric Lander and John Sulston over in the UK, Bob Waterstein, Shirley Thielman, and many others. Now, the eventual cost here for this, what was originally slated to be a 15-year project, was $2.7 billion, so about the price of one of those nice shiny new nuclear aircraft carriers out there in San Diego Bay, for example. Now, Craig Venter, who many of you will have heard of, he was a graduate of our program, of course, at UCSD, and originally the Human Genome Project team, the Tiger Consortium, and actually worked intramurally at the NIH in this project. And he kind of pioneered and pushed for a new sequencing approach called shotgun sequencing that we'll mention in a moment. And there was a rather public and well-documented falling out and disagreement, and he went off and started a private company called Celera Genomics. And this, in 1998 at this stage, this started a really high-profile race between the public consortium and the private sector led by Celera, where they were really trying to use it as a business model to patent or patent genes, genetic information, for future uses. So a company that had that information, they would be the ones that were able to develop new pharmaceuticals, for example, new chemotherapeutic agents that would go after those genes, for example, solely. So this was a race that the public consortium just could not lose, right? And in the end, rather amicably, to the surprise of many, actually including myself, both initiatives in this race released their initial drafts early. This was in 2001. There was Craig Venter and Francis Collins on either side of Bill Clinton at the time at the White House, and talking about this first draft of the human genome in 2001. 3.2 billion base pairs. Now, slightly refined numbers, 20,400 coding genes, about 24,000 non-coding genes. And simultaneously over in the UK, you had the satellite link with Tony Blair, the then Prime Minister, with John Sulston, and lots of other scientists like Fred Sanger, for example, there, along talking about this Herculean effort and an achievement at that time. Now, what was the big deal? What was the shake-up that Venter and colleagues and others were kind of introducing to propel this, 5 years early, this completion? Well, it was shotgun sequencing. So what I've got here, if you bear with me for a moment, is a video. It's not available on YouTube, unfortunately, so I'm going to show it here. This is from Howard Hughes Medical Institute, and it describes a little bit, or gives you a sense for shotgun sequencing and how different it is to those approaches. So bear with me for a second while I start this playing.
Shotgun sequencing is the method that was used by the private Genome Project. Shotgun sequencing requires multiple copies of the genome, which are effectively blown up into millions of small fragments. Each fragment is then sequenced. The small fragments are assembled using an immense amount of computer power to match overlapping sections. The drawback of this method comes when dealing with repeat sequences. Often there is no way of knowing how long the repeat sequence is, or in which of the many different possible positions the fragments overlap. Even the incredibly powerful software used to shotgun sequence the human genome couldn't cope with this. So Celera, the private company which relied on this approach, had to use the public data to fill in the gaps left by the repeats.
Celera, tomato, call it what you will. They were public enemy number one back in those days. I actually worked in a company called Decode Genetics shortly after this. They were in the same game, sequencing human genome, in that case in Iceland, right, where there was a homogeneous population. They could trace their people back to the first settlers, very little immigration, a real hotbed for discovering genetic diseases. And they had a room just like this one, shown here in the video from Jonathan Wiseman that I mentioned earlier, that talks about these sequencing approaches in more detail. This kind of industrialized approach to science that was very foreign to molecular biologists at the time and to geneticists, right, where there was technicians and these ABI kind of capillary sequencers, these big rooms running 24/7 around the clock, churning out sequence, right? It was really a race to get the sequence and put it in the public domain at that time. So, refer to Jonathan Wiseman's video there if you want to delve into more about these equipments and the technical innovations that really enabled this project. They really are quite remarkable, as we'll see in a minute when we talk about costs of these things. But what I want to highlight and make sure everyone's on the same page is next generation sequencing technologies, what we call NGS approaches for short. They really resulted in this big shift away from those long reads with low coverage, right? This kind of first image here, to these short reads. So many short reads at high coverage, right? So we've got many reads for a given region here of our reference sequence that overlap it here. And this provides numerous opportunities, providing you have the computer power, as was just discussed, the bioinformatics chops and know-how to analyze these things, right? That had to be invented too here, that allows these expanded kind of genomic applications. So what made this all possible, of course, is that rapid progress in sequencing technologies, both in cost effectiveness, as shown here in these curves that you'll see all over the place in these kinds of talks, right, the Carlson curves, if you will, but also in the effectiveness of these approaches, right? So here, this 20,000-fold change when Solexa came along, and they were bought by Illumina, of course, here in San Diego. This huge change and decrease in cost has put these things now, right, we're here at the end of this graph around this $1,000 mark or slightly above it, depending on which press release you believe. This puts this in the ballpark of things like MRI, right? If you're lucky enough to go to a hospital and your physician orders an MRI, it's going to cost the insurance company or you or someone around $4,000 US. And a genome sequence, a whole genome sequence, is going to be around that, if not less, with current techniques and current technologies. So this opens up a huge new window on using genomics in biomedical sciences, right? Not just for rare disease diagnosis, of course, where if we can find the genetic basis of these diseases, we can have a diagnosis and hopefully a tailored treatment, but also in cancer therapy, metagenomics, right? The sequencing the microbiomes and things like this, right? But primarily cancer genomics, as we'll see in a later segment of our course. Really revolutionized by this approach to biology, this genomics approach. Now, if I was given this talk just a handful of years ago, I would have talked about two approaches: sequencing by synthesis. This is the Illumina approach, and I would have also talked about an alternative approach that doesn't use a polymerase but uses a ligase, so-called sequencing by ligation. But really, there's no need, you can see it's grayed out here, to talk about the sequencing by ligation, because Illumina has won this battle. Sequencing by synthesis, using a polymerase to incorporate and assess nucleotides by a primer one at a time, is now the be-all and end-all, really. It's the dominating technique in the market. Illumina dominates this sequencing market. In fact, today, I think it's well over 90% actually of all sequencing is done on Illumina machines. Now, they generate millions to billions, so huge numbers of high quality, 99.9% accuracy, but short, so on the order of 300 base pair reads per run. Okay, your accuracy is good as if you have short reads, as we'll see and maybe discuss in our hands-on session. The numbers actually, the numbers of, they're kind of dependent on which machine you have, what kind of type of machine you have. Just like you can have different types of cars, different models are more expensive than others. It's the same with sequencers here. We have the kind of desktop sequencer, the MySeq one that we would, if we were in our face-to-face course, we would just walk around out of the classroom and go behind the classroom and go see one of these MySeqs that we use for teaching in our teaching lab. They generate about 30 million reads. They cost about about $100,000 currently, and if you wanted to buy one of these off the shelf at the minute. Then NextSeq's in the middle here, about 3 billion reads, often used in many lab research. Individual researchers or teams of researchers will use these things. And then the big machine, the NovaSeq. These images aren't to scale here. They'll be the size of a big freezer, right? Fridge freezer kind of combo that you would have. They're the big players that generate 13 billion reads. Currently, you won't get much change from $1 million if you want to buy them today in the current year. If you believe the press release and you run these things multiplexed to the nines and run them around the clock, you can get a human genome in 48 hours on this biggest machine for around $1,000. And that's a game changer, right? This mythical kind of $1,000 genome that people are pushing. Now, if we were in the hands-on session, I would pass around, as well as going to see these machines, we would pass around these things, right? These are the flow cells. Here's the kind of NextSeq, and here's a little MySeq one, and others. So these are microscope cover slips. They're glass, and they have regions in them that you wash your reagents into, both your sample, your DNA, but also the reagents the machines use, right? These things. These are Illumina flow cells, and they have different capabilities depending upon which machine you're using. The flow cells are machine specific, of course. Now, the idea with all of them, of course, is we have our DNA to be sequenced. We'll call that an insert in Illumina speak, and it's have adapters that are added to it, so specific sequences that are added. These are both primer binding sites for the polymerase, of course, but also capture sequences that will allow your sample to stick to your flow cell, right, and actually undergo the sequencing approach here. So this, what we call our library, when we've got these things, all adapters on, that'll go in, it'll be washed into your flow cell with these reagents. It'll be complementary to those adapters that are on the flow cell. It'll stick there. It'll undergo what Illumina call bridge amplification, this kind of bridging across here that you see in all their images and documents. This is the purpose here is to concentrate that homogeneous little sequence there, right? So we'll have clusters of the same sequence on our flow cell, many of these. And then when we've got enough of them, we'll be able to detect them. So it all starts with a single kind of molecule, but now we've got large numbers of these molecules in this cluster that we're going to take into the second of our 3 steps, which is really the kind of critical one here, where we're going to add polymerase mediated addition of fluorescently labeled nucleotides. So same as those old techniques, right? You've got your 4 bases and they've got different fluorophores on them, different wavelengths they emit. Here I'm coloring them up like Illumina does. They're not really red and green and yellow and blue, but they're different wavelengths. But the idea is they're labeled and they're blocked, so you can only add one at a time. But you can wash them off. You can wash the fluorophore off, so you're not going to get complications when you add the next one. And you can also wash off the blocking group as well. So then you can do repeated cycles of washing in your all 4 nucleotides and seeing which nucleotide binds to these clusters based on its fluorescence. And we actually monitor that in the 3rd step, the emission from these incorporated labeled nucleotides is shown here. So what's shown here are these little panels are like the images of a movie, if you will, so different iterations of adding these blocked labeled nucleotides here. So in the first one, in the first panel, if you concentrate on that first dot, you get a green signal, right? That wavelength is lit up, so we know there's a C at this stage. Then the next cycle, we get a different color, we get an A, a blue, etc., etc. So we're going to build up the sequence step by step by step by step. But of course, it's not as beautiful and clean as is shown here in these kind of images that I've taken from this paper and adapted here. Biology and indeed chemistry never works 100% just the way you want it to. So there's always going to be some of those dyes that aren't labeled, or some blocks that aren't going to work, and you're going to get out of phase, and you're going to get various problems as you lead up, which actually leads to this length limitation of these Illumina approaches, this 300 base pair kind of limit that you get. And we'll talk more about that when we actually start analyzing the data and seeing how the quality drops off as the reads get longer. Now, if you want a more coherent description and a lot more tech speak and jargon speak introduction to how Illumina sequencing work, you can go visit this video here from Illumina themselves. They'll talk all about bridge amplification, about their proprietary approaches, and how these things work, along with little animations that are quite good at explaining how the Illumina approach overall works in a big picture kind of scheme. So I encourage you to do that if you're so motivated. What I want to make sure whether you do that or not, I want to make sure we all are aware of some terminology that's going to be important for a hands-on section in this field. We're going to talk about insert size. Of course, the insert is the bit of the sample that you're sequencing, and there'll be a range of these, but there'll be some mean or size here, and that's our insert size. We'll need that in our bioinformatics. It'll help us if we know that information, because we often sequence from both ends, and if we know how far apart they are, that'll help us when we go to align them back to a reference genome, for example. We'll also talk about sequence coverage. So this is, again, how many of these reads map or align to a given region of your reference genome on average. So here in my little graphic, I have 6 of these little blue lines here. These are the reads mapping to this black reference genome. So we call it 6x. We typically work on many, many more x. And the more you have, of course, the better. The more confident you're going to be about calling differences. What is variance, real variance versus errors in the sequencing, for example. Now, another key piece of terminology is the generations of DNA sequencing. There's the first generation approaches that we talked about, second generation approaches. This is the Alexa or Illumina approach that we just described. But then, there's other approaches, right? Typically nanopore, single molecule approaches, and folks will call them third generation. So we have a bit of a terminology problem here. It's not like Star Trek where we have next generation and then next generation, right? We call it kind of third generation approaches. Now, it's kind of hard really to come up with a consensus about what these third generation approaches are, because they're still really in active development. Typical characteristics of those that are available are long reads, typically in the order of 1,000 base pairs or more. That's great, right? That's going to make the bioinformatics much easier than these smaller fragments when you go to put your jigsaw back together. Single molecule, this is also great because we don't need our amplification, PCR amplification steps anymore. And they're often associated with pores, nanopores kind of technology, where we have maybe the detection of a current change as the nucleotides pass through a pore in a membrane, for example. Things like Oxford Nanopore, the MinION sequencer that I'm showing in the picture here. Right, this is a small handheld sequencer that's USB. You can plug it into a USB drive on your computer. You'll see these used in the field. You'll see scientists out in West Africa taking Ebola samples, putting them into these things, or in the space shuttle, not the space shuttle, but the space station, for example. You'll see these MinION sequencers being used by the astronauts and whatnot. And I don't want to say that this is the only approach. There are many others being developed, but it's kind of, no one really has them in their hands yet, so it's not really clear what kind of characteristics they're going to have. There's always a lot of hype and a lot of promise, but until we actually get them and see them and how they work, it's hard to really define what their characteristics are, of course. Now, this is a very fast-moving field. For example, at the end of last year here, there was, this is from Oxford Nanopore and collaborators, there was the first direct RNA sequencing by nanopore, rather than doing Illumina RNA-seq, where you have to, of course, you're doing DNA sequencing, so you have to do cDNA, you have to convert it to cDNA and do all these sorts of things, right? Here we could do it directly, or they could do it directly. So I've placed the abstract here. For this paper, I'm just highlighting a sentence from their abstract that really highlights the promise here. Current methods, that's the kind of Illumina approaches, they have limitations due to those short reads and the reverse transcription and the amplification biases that are going to be introduced depending on what enzymes you're using. Of course, here with this direct nanopore approach, you skip all of those. It really shows great promise for doing these things better than we could do them or can do them currently. So it's definitely an area that's developing rapidly. So before we stop here, I just want to mention, what can we do with all this sequencing information once we get it, this large-scale sequencing information that we're accumulating. Next video, of course, we're going to talk about how do we analyze it, but what can we do with it, right? Well, what we can start to do now is population scale analysis. We don't just have one genome, we don't have Craig Venter's genome anymore, right? Or one another person, or the handful of people that were sequenced with the Human Genome Project that's not representative of the diversity within our class, never mind the diversity within the population, the world population at large. But once we do have a large number of samples, as we're accumulating from these projects that I'm showing the kind of splash screens or the icons for here, we can start to do cool things. We can start to do real population scale analysis. We can start to assess genetic differences on this large scale, both naturally occurring variations within human populations and non-human populations, what makes us distinct from other animals, for example, as well as disease states. What's somatically within tumors is a classic example where this has really changed the game, right? It's not, tumors are no longer classified solely based on the site of origin, but on their genomic features. What are the mutations that actually are present in these tumors and driving them and actually druggable potentially in these tumors? This is a real game changer in this field. So just like the old quote, variety is the spice of life, in genetics and genomics, variation is the spice of life. So as I said, having the sequence of a single white Caucasian male or a handful of them was great. It's not the be-all and end-all. It never was and never will be. It's not representative of potential differences that can occur between individuals. These are the very things we want to find out. What are these variants? They could be the cause of phenotypic differences, such as different morphology or susceptibility to disease or pharmacogenomic features, the effectiveness of different drugs. These are exactly the things that we're able now to study as these sequencing approaches ramp up. So I'm going to finish there, and when we come back, we're going to start looking at what the data looks like that comes from these sequencers and how to analyze it. That's our next video in this series. So join me then, please. Thank you.